start. Good afternoon, everyone. I'm happy to be here today to talk about microbes in food. So before starting, people who don't know me, Rangel. I am a course instructor. Why I'm here? Because I'm a food scientist. Working in the Department of Nutritional Science, but I'm a food scientist, and I consider myself as a microbiologist. I hold a PhD in food science from Laval University and a master in same. And then all my grad studies, my postdocs, and part of my research work in Dr. Comelli's lab, I was focusing on microbes. So either microbes as a microbe itself, either in the food matrix, or microbes in interaction with the host. But today I'll be mainly talking about microbes in food. And the reason why I'm here also, I worked with Dr. Comelli for years. So I started working in her lab as a postdoc. Then I was hired as a research associate, working in the same lab. And then I was hired as a course instructor. So I'm mainly teaching. I'm no longer working in her lab, but we're still collaborating and we still have very interesting work to do together. Food, why microbes in food? So we know that foods are not sterile. We're exposed to microbes every day, on a daily basis. Microbes are everywhere. Nothing is sterile. Even blood, there are studies showing that the blood is not sterile. Fetuses are not sterile. But this is not the topic of today. So, there are different sources of microbes. So, primary sources can include soils, water, include plants, plant products, etc. And then microbes, there are the bad and the good and the ones in the between. This is what we find in food. The bad ones are the ones that are responsible for foodborne illnesses, food and intoxications, etc. And the good ones are the ones beneficial, so the ones that we are using for fermentation, to enhance the quality, the probiotics, etc. These are the ones the good one. And the one in the between, these are we consider as opportunistic. They are here, they cause spoilage, etc. They're not really harmful, but they are not the one that we want in our food. So we'll go over these types of microbes in this lecture. So today I will be giving you an overview about what factors influence these microbial growth. What do they need to grow and how do they grow? So, because they don't grow the same way, they don't have the same need for nutrients the same way. So microbes growing on meat are most likely different from the one growing on cereals, etc. And then we'll go over microbes, the microbial spoilage, and I will give you a brief introduction on the quality control and how we do it. I did hide some slides, so they're just to cut from the lecture, but you guys have them. And then we'll talk about food preservation. The last part, I won't expand it a lot of time, but I would like to share some information about what is done in terms of new genomics and the new research and what can be done to improve the quality of food, etc. So these are the topics of today. We're here to learn together. You'll be learning something probably new, and I may be learning something from you as well. So feel free to interrupt, ask questions, any question, I'll be happy to answer. So what are the factors influencing these microbes? Why do they grow? Microbes in general, they need a source of sugar, they need a source of protein, and they need, of course, a source of energy to grow. And then There are different factors within the food matrix, within the food, that help these microorganisms to grow. So when we talk about the food itself, intrinsic, we'll be talking about composition. So composition, food that is rich in sugar will likely help an increase or overgrowth of microbes. We'll talk about water activity. Just this morning we spoke about it, 86 people who are here. AW, water activity, it's the water that is available for microbes to grow. So higher water activity meaning higher chance for microbes to grow. Then the pH. Most of microbes do not like pH, do not like lower pH. So this is why acid, most of them, they don't grow in acid. So it's one of the factors. The presence, absence of oxygen, presence and absence of antimicrobials. Then when we talk about intrinsic, outside the food matrix, so outside the food itself, so we have oxygen. You may have seen this last week. So there are microbes that are considered anaerobe, so they don't like oxygen, they don't grow in the presence of oxygen. Others, they are called aerobic, they like oxygen. So these can be an important factor. Temperature, so 37 degrees, most of microbes grow at 37 degrees. Others, they are more...
Was it Celsius?
Yes.
Okay.
Some are, we call them psychotroph, they can even grow in the fridge, they grow at lower temperature, etc. And then relative humidity, talking about extrinsic and the atmosphere. Process, it depends on the food or how the food are processed. Washing, handling, all these are factors. And then together, these factors will help or affect the synergism or the antagonism of microbes. So they help each other to grow, or they compete with each other to grow, and which affect the growth rate. So how this happens? So usually we have microorganisms that are present here, and then we have intrinsic factors and extrinsic factors. First, we need microbes to grow. Something cannot grow they cannot grow. If there is no microbe, there is no growth, of course. So here, all these, they help an overgrowth or an activation of growth. And then, if you go over time, if you change on time, so these microbes will grow. They will create a microbial community. And then this microbial community, because they are producing metabolites, they change the pH, etc. So this will change either the extrinsic factor, and will also change the intrinsic factor. So if they may produce CO2, and they may produce acid, so they lower the pH. They may produce the CO2, so the product will, it's a gas, so the product may extend, etc. And then all these will lead to a food spoilage. So what do we mean by food spoilage? So it's a process of deterioration of food that makes it indesirable. We don't want to eat it. So it alters the visibility of food, so we don't it changes the color, it changes the organoleptic, and spoiled foods are generally not toxic, so they lead, or they can, but so they're not, people don't die with spoiled food most of the time. So spoiled food are considered as unsafe because of the number of microbes that they're overgrowing, and then which indicate that food pathogen may be here. So food pathogen, the pathogens may be present in spoiled food. So there are different types of spoilage. There is microbial spoilage and there is a non-microbial spoilage. Today we'll be focusing mostly on microbial spoilage, the ones that are caused by microorganisms. So what do they depend on microbes, obviously, and they depend on the factors that I cited. So presence of nutrient, these are intrinsic factor of the food. pH, AW. And then we have common microbial groups or groups or microbes in general that are commonly used or commonly found in spoiled food. So, for example, Pseudomonas, this one. Pseudomonas is you can find it in meat, spoiled meat, milk, etc. We'll see in a few slides. And then we have cyclotrophic bacteria. This one, it can be more dangerous because they multiply, they grow, and they resist to a lower temperature. So they can multiply in the fridge. So this is why, for example, we keep food in the fridge, and then after 1 or 2 weeks, and we see that the food is spoiled, and we cannot consume it anymore because there was no growth. And then we have spores or endospores. These are more complex or more dangerous than these ones. Why? The spores, these are vegetative forms of the bacteria. They resist to the heating and to the pH, so they are more resistant. And then we usually find them in canned foods and non-processed in canned foods. So they resist, and then when we start heating the food, and if it's not heated properly, we have this, it's a dormant microbe that starts regenerating or resuscitating and overgrowth. And these can be very harmful because they produce toxins that can be harmful. So Clostridium,
Sorry, you said so it's dormant and the heat will cause it to reactivate?
So it's dormant, but then when you heat it, for example at 55 or 58 or higher than 50, they start growing. If they are not, so for example, if you have canned food, and then it's already contaminated, it has a spore, the spores were not eliminated. Then you heat it, but you don't heat it properly, there is a higher chance that these spores start growing, and then the person gets intoxicated.
But you can heat it enough so that it's like...
So if you heat it enough, yes. Okay. Yeah. But they are very resistant, these ones. Okay. So then we have fungi that can grow in less favorable. So the fungi, at the opposite of bacteria, they grow in dry with even lower AW activity of water, etc. And we can find them, so these are mostly contaminant of cereals. Yes?
You just mentioned that when you were conversing with the other student, you said if you heat it enough, it will be fine. When you say if you heat it enough, even the endospore forming organisms can survive it, can die if the cooking temperature is high enough?
The heating at home, you don't really kill the endospores, but you don't allow it to overgrow. It's like you may have the endospore. Endospore as it is, it doesn't do anything. Because it's a dormant. It's like something inactivated. So even you ingest it, it doesn't do anything.
So then, is it when you heat it to greater than 50 degrees, that little bit of temperature is the one that is waking it up?
Yes. Yeah.
Okay. But then if I were to, instead of heating it to 50 degrees, what if I heat it super high, that it wouldn't have, it would stay dormant?
It will stay dormant. It doesn't, unless you heat it at 100, you boil it for 10 minutes, then you're sure that you're really destroying it. So if you heat it just a little bit, it's just like you warm up. So you warm up your meal. Here you're increasing a chance that you warm up the meal and then you leave it on the counter for 5, 10 or even an hour and you consume it. This is no. You heat it, you put it on the stove, I'm coming back. You put it on the stove and for 10 minutes and it really boils, this is okay. You consume the canned food as it is without heating, with spore that is dormant, it doesn't do anything because it didn't have the time to resuscitate before you ingest it. Because the spores, as they are, they're not toxic. But the spores, when the bacteria is resuscitating, they produce toxins, and the toxins are the ones that are harmful, not the bacteria or the spore itself. Did you get it?
So the spore itself isn't toxic, it's the organisms that it can form?
Yeah.
Okay.
So is that like syntropy or syntropy that we talked about, where it's like the spores are promoting the growth of other bacteria?
No, the spores, they grow, the spore is a dormant, it's like a microorganism that is inactive.
Okay, so do we like face the same risk or like lack thereof when we eat it cold versus when we eat it really heated?
So,
You're surpassing the...
yeah, no, the risk is not the same.
Okay.
Yeah, so the spores, you consume spores that are inactivated, no, they should be fine.
Okay.
But the spores that were not hit, it's like you shouldn't give them the chance to survive, to be active.
But if you heat it enough, like it's the same risk. If you heat it and like past that threshold of like killing all the bacteria versus if you were eating it cold. Like there's a sweet spot.
Yes,
There's an
so you don't have to allow them at specific, there is kind of a range. So no risk or less risk,
Yeah.
high risk, and then no risk.
Okay.
So you don't have to stay within this range. Very high, that is fine. Cold is okay, but within the range is not fine.
I've got a question regarding the definition of endospore. So if I Google it, like a spore living in the cell of bacteria.
Yeah, it's a spore that is inside the bacteria.
So it's only in like those two bacteria, and then the spore stays inside the bacteria.
No, the spore is just in like Clostridium bacillus. It's mainly in Clostridium. You don't find spores in Pseudomonas or other.
Okay. So
No.
that's
These
only-
are specific to these two examples.
Okay.
And the one that we really find it in the food is the Clostridium. Bacillus, but Bacillus is less likely to be found in food unless it's contaminated by soil, etc. But Clostridium is a very common food contaminant.
Thank you.
Okay, so what else? Then we have non-microbial, so we're not covering this today. So I'll be giving you a few examples of food spoilage. So the milk. So why milk is susceptible to bacterial overgrowth? Because if you remember, we said in the beginning, we said there are intrinsic and there are extrinsic. So intrinsic factors of milk is the AW. AW is the activity of water. The water that is available for microorganisms to grow. And then AW higher than 0.7. So the AW range from 0 to 1. 1 is water. So water has 100% of water is available. Then we have liquids, so 0.98, etc. These are milk. So milk has an AW of 0.98. Then it has a pH around neutral pH. So this is the favorite pH for microbes to grow. And then it's rich in nutrients. So all what microbes need, sugar, proteins, everything. Milk has its own microbiota. And these microbiota, we'll just give you a few examples later. So these microbiota or microbial communities, same as for gut, they are specific to farms. There is a core, what we call core microbes. So you will find Pseudomonas is everywhere. Every farm here in Ontario, farm in Quebec, farm in Alberta, wherever, it's the same. And then some microbes are farm specific or season specific, etc. This is just to give you an idea. So, what do we find in milk? We have lactic acid bacteria, we'll see what is this. We have Pseudomonas, remember? Pseudomonas, you see them, the name is everywhere. Then we have Staphylococcus and we have the yeast. The milk itself in the mammary gland is sterile. But then it goes up, there is a contamination from outside, from the animals, we'll see in a few slides, from the environment, from the grass, and the farm, etc. So these are the source of contamination. So milk spoilage mainly involves psychotrophic microorganism. Pseudomonas can be a dominant one, because usually when the milk is collected, it's refrigerated, is kept in a fresh space, so it favorizes the psychotrophic microbes. Pseudomonas is known to produce heat-stable proteolytic enzymes, and even lipolytic enzymes. So these enzymes, they deteriorate. These are lipase and protease, so they deteriorate the fat and the protein in the milk, so which deteriorated very fast and decrease the shelf life. Then we have meat. So meat, it depends on the. It depends. So meat, as itself, is supposed, or muscles are believed to be sterile. This is before. So live, for example, live animals, they have sterile muscles, or until they are. They become carcasses, so they are killed. And then this is where the contamination comes. So contamination comes from outside and from the gut, from the animal's gut. So meat composition, why the meat is one of the foods that are easily spoiled? Because water composition, so it has around 60%, an average of 60% of water, has a protein, perfect for microbial growth, carbohydrates, vitamins, etc. And it has the AW rounding 0.97 and 99. And you would say, yeah, but this is close to milk. And milk is liquid, and then the meat is not liquid. Because the AW is the water that is available for microorganisms to grow. So it doesn't necessarily mean the percentage of water or the water content. So we have to make a difference between water content and water that is available for microbes to grow. So here, this is why they are closer. So this is why meat is a good. So we can find. So this is from an old reference, but we still have seen the same again and again. And then you can. These are the common microbes. Sorry. Yeah.
So 0.7 is like the AW.
0.97. 0.7? Yes.
When bacterial growth starts, right?
Microbes, mold and yeast. Bacteria is 0.8 to be exact.
Okay.
So, but to avoid all the microbial growth, microbes in general, yeast, etc., we need to have an AW lower than 0.7.
To prevent all microbes.
Yeah. Yes. This doesn't allow microbes to grow. Okay, so these are the lists, so you can see. And then you can see, for example, there is a difference between the fresh, so microbial content or microbial presence, there is different from the fresh and the. So meat spoilage can be different, so it depends, is the fresh, spoilage of fresh meat is different from the spoilage of packed meats, for example. So here, so usually red meat, so it's exposed to high level of oxygen because it's stored, chilled aerobically, so in aerobic condition. And then here it produces high redox, so high oxygen levels surrounding the meat. This environment, so we're talking about extra and sec factors, these they favorize the growth of gram-negative microorganisms. Pseudomonas is the first. So Pseudomonas is like everywhere. Acidobacter and Enterobacteriaceae. Enterobacteriaceae we talked about, for example, E. coli. So these can be one of the contaminants. And then this is kind of a cascade. So we have Pseudomonas starting, and then other microbes taking over, and so on, etc. And then these microbes, when they grow, they produce metabolites such as ketone, volatile ester, etc. And these are the ones that are responsible for the off flavor and the odor of spoiled meat. So because of the metabolite. So how this works quickly? So there are steps. As I said, there is a cascade. So first you have Enterobacteriaceae that are producing a different metabolite. We have alcohol. So these are alcohol producers, acetanoid, and then these are responsible for a flavor, a buttery flavor. This is like kind of a flavor that is a mix of alcohol with weird, I don't know how to describe it. Then we have Pseudomonas that are producing ester, as I said. And then Pseudomonas are the one, they're not only responsible for producing the off odor, but they are the one that are, they can also be responsible for the odor and the flavor. So for the petrification of meat, so changing color and odor and this weird one, it comes from mainly Pseudomonas. And then all these depend on numbers or charge, or what we call microbial charge. How many microbes that are present within, for example, a centimeter square of milk, or within 1 milliliter of, because when we set the norms, so for example, for quality control labs, etc., there are norms. And then we say, for example, salmonella, we should have 0, salmonella is a pathogen, we should have 0 salmonella per 25 gram. This is a norm. Everywhere you go, it's like this. So it's 0 micro per 25 grams of meat. Pseudomonas, there should be less than 1,000 or 10,000 microbes per centimeter square, etc. So this is how we can say, okay, there are microbes, but they're safer to go, safer to be processed, etc. This is just for your information. And then when microbe in the meat, when microbe reach 10 to the 8, which is too much, and then here it becomes visible. It's not only for the odor, but the color changes, and it becomes like kind of viscous. So these are the metabolites that are produced by these microbes. So as I said, yeah.
By the term visible, do you mean like we can see like the microbes, or
Sorry?
by the term visible, do you mean like we can see the microbes?
No.
Like they're
No. We see the metabolite, we see the transformation, because it becomes kind of a viscous on the surface.
Okay.
It changes the color, and then so odor obviously, but the color changes, and then when you touch it, it becomes kind of viscous, gluey. So these are coming from the metabolites. Okay, so this was about fresh meat, and then vacuum packed or modified atrazine. So these are the ones that we find in the grocery store, packed, either vacuum one or within a modified atrazine. Just to say something. So when we find so when you go to grocery stores and you see them, and sometimes they're too red, like it's red flashing. So these are because of two reasons. So it's either the package, because they modify the atmosphere, and they expose it, so the lighting of the meat is different. So the lighting, when the light, so on the shelves when they have meat, etc. So the lighting, so usually they play with the lighting, and then because it's packed in modified atmosphere, so this is why we see it very bright, red bright, and you take it home, you open, and it's not really the same color. So, okay, so you remember the fresh one? There was I'm checking because sometimes I get too excited and keep talking and I forget myself. Okay, so vacuum. So we do a vacuum, we remove the oxygen. here, so we remove oxygen and sometimes we purge the CO2 when we do a vacuum. So there are two ways. So either vacuuming with nothing, but most of the case vacuuming and purging the CO2. Either way, yeah, CO2. Because of the CO2 here is present, and because meat is not sterile, so even before putting the meat slices, for example, in the package, they were not sterile. So there were kind of a contamination, although the number of microbes didn't reach high level, but still, they were not sterile. Then, microbial community of meat contain lactobacilli and leuconostoid. These are, we'll see them, these are lactic acid bacteria. They take the glucose, sugar, and they produce it in lactic acid. So, and so they, yeah.
When it says sour odors develop, is that like right when it's packaged, or is that if you leave it for a long time?
No, if you leave it for a long time. Yeah. So the sour, because we're producing lactic acid, so it becomes more acid, it's sour product. So the responsible are these ones. Modified atmosphere, because we similar to the vacuum, so we favorize, yes, no.
What's G plus?
Gram positive.
Oh, oh right.
Yeah, G1, G minus is the gram negative and G plus is gram positive. So, same modified atmosphere, so we have the same thing. And then we favorize homofermentative, so we'll see it in a few slides what are homofermented. So these homofermentation, they produce lactic acid and CO2, so two compounds from the fermentation. So this is just to give you an overview about common defects in meat, so what do they cause, and what are the bacteria that are responsible. When you think of, yeah.
The heterofermenters also make CO2 and lactic acid, but it's less, right?
Yes. And the first one, this one, the CO2 is the one purges. This is what favorizes. CO2 here, it favorizes the growth of anaerobic. And then when these one grow, they start making the lactic acid. Yeah, so these are the, yeah.
Sorry. For the modified atmosphere, you mentioned about like the shops, like different lighting. But I mean, even if the lightings are different, it's still like with oxygen. So I wonder why it's still the same as the like the vacuum.
I didn't sorry.
Oh, sorry. I mean, the meat in modified atmosphere, as you listed as like shops, I think they're still having oxygen, so I wonder why they are having like a similar situation to vacuum time.
What do you mean by the overgrowth?
I mean, they are having oxygen, so I
No, when you pack, you remove the oxygen.
so what
You remove the oxygen from both of them.
okay, so what does the modified atmosphere?
To modify the atmosphere, you remove the oxygen and then you put a mixture of gas.
Oh, okay.
Yeah, yes, this is the modified atmosphere. So you remove, in both cases, you remove oxygen. Okay, so how about cereals? Cereals, you will see, you might have noticed that there is no pseudomonas. So cereals, they have their own microbes, contaminant, etc. Because, so cereals, they have lower activity of water, and they are, so, and then the contaminations are different. So we have either the filter, these are mainly molds, so we have either the filled mold, or we have the molds that are overgrowing during the storage. So for the mold, we know the fusarium, it adapts well to all the conditions. Aspergillus or penicillin, these are the common ones. They can adapt. The reason why, because they adapt to different conditions, different temperature, etc., and they can grow even at lower temperature. So when these molds start growing, so like they respirate, so they produce gas, etc., and which will lead to an increase of the AW. So going from 0.4, 0.3 AW, and then the overgrowth, they make them produce or make the intrinsic or the cereals themselves more wet. So it increases the AW, or the water will be more available, which is a good or favorable condition for growing. So it's kind of a cascade. So the molds start growing, they make the condition favorable for other microorganisms. Or what is not really wanted for this overgrowth is the production of mycotoxins. So these are toxins, natural toxins, and the production of very specific odor of these cereals. This contamination, yes.
Is mold different from, they're different from microorganisms?
Yes, but they're different.
Okay, what's the difference?
So the difference is their way of growing and their needs. So they don't need the same, they can grow at the opposite of microbacteria. They grow in non-favorable conditions.
So molds aren't microbacteria?
They are categorized as microorganisms. Yeah.
But they're different from...
They are different, yes, of course.
Okay.
The size is different, the metabolism is different, everything is different.
So even an AW lower than 0.7 can prevent the growth of all the microorganisms, but not the mold.
The mold, they can grow at 0.7.
Okay.
At higher 0.7 and higher, the mold can grow.
The lower is like still. Like
They can grow at lower, yes.
Okay.
They can grow. Sorry. They can grow. They can grow at lower, but if you see the chart, so this is kind of a chart for AW, like a rule that everyone knows. It's kind of an international chart. And the chart tells you 0.7 is the limit. But there are cases where there were studies where they found that specific strains of mold, they can grow at lower AW. Okay, so, and yes, so, and then the one that is more known is the aflatoxin.
What exactly is the mold doing to increase the AW? Like, what's the process?
So they produce, they change, because they change the composition of water, they change the composition so they produce metabolites, they may produce CO2, and they did create kind of an environment to make the water, to increase the moisture and to make the water more available.
But it's not changing the amount of water.
It doesn't really change them. It may increase a little bit content, but it doesn't really change. They don't get really wet and you can see it. No, it's just the environment that they are creating. This is what it favorizes the changes of AW. Okay, so this is kind of a picture of the spoiled corn. So how about foodborne illnesses or foodborne diseases? So these are commonly referred to as food poisoning. So ingesting food and we get sick, so we have a food poisoning. So then there are two types of foodborne illnesses or food poisoning. So it depends on what we are ingesting. Are we ingesting the microbe or are we ingesting the toxin that is produced by microbes? So depending on the type, we call it, by definition, we call it foodborne infection or foodborne intoxication. Infection, this is what we ingest, live microorganisms. Live microorganisms, so we consume them. by the time that they reach the gastrointestinal tract, usually they infect the colon, so they either produce the toxin or etc. So it takes time. They take at least 24 hours or 1 day. And then after 1 day of ingesting these toxic microbes or microbes, and then the symptoms start appearing. Diarrhea, cramps sometimes, etc. And then the common one are the Salmonella, Campylobacter, and E. coli. Escherichia coli, this is a serotype. So Escherichia, we all have, most of us, have E. coli, Escherichia coli as a part of the gut microbes. But they are different. So the pathogenicity is strain specific or serotype specific. So not all E. coli are pathogen, but these specific ones. So these are, they can be found in different types of foods, but they are known to be a contaminant of meat, ground beef, etc. So these ones. Campylobacter is very resistant to antibiotics, and then these are known to be contaminant of chicken. Salmonella, it can be everywhere, in fruits or vegetables. So these, if they are ingested, the symptoms start appearing after 1 day. Then when we talk about intoxication, so for example, we ingest the Clostridium that was in the canned food, that was not heated properly, started growing and started producing toxins. So we ingest this canned food containing, containing Clostridium. Because the toxin was already released in the food, the symptoms start appearing within few hours. So once she's rested, they appear within 3 or 4 hours. Because the microbes do not need to grow, etc. We have already ingested the toxins. And the one causing these intoxication are Staphylococcus, Bacillus, and Clostridium. So the most common one is the Clostridium. Okay, how do we prevent this? So we should prevent, so everyone is responsible for preventing these foodborne illnesses. So farmers or the producers, manufacturers, the ones that are processing the food, and we as the consumers. So here on the farm, so what can be done? To reduce, we cannot sterilize obviously, but we need to reduce the number of microorganisms. So for example, Salmonella, if you remember, I said as per the norms, international norms, we need to get 0 Salmonella per 25 grams of food. This is the rule. So on the farm, farmers are responsible for testing for Salmonella. They're responsible for minimizing rodent and birds, etc. These are the main source of contamination. And they're responsible for isolating new animals. In Canada, there was a program, an approach that was called On the Farm Food Safety. So establishing what we call HACCP or HACCP, that is Hazard Analysis Critical Control Points. So every single point in the farm, in the slaughter, etc., should be controlled. So we should identify what we call CCP, critical control point, to see what is the source of contamination and how these source of contamination can be controlled. So these are what we call the HACCP or HACCP. And then during the processing, preventing food contamination using like strict personal hygiene. So this is because contamination may come from the personal. So usually it's a very strict. So we avoid what we call cross-contamination. So contamination. So in the industry, so usually the way, for example, there is an entrance door of contaminated food, and there is an exit. So we should never go back, etc. There is kind of a process and path that should be followed. Controlling temperature is very important. Cleaning and decontamination. Then at home, we're responsible for washing defrosting meats in the refrigerator to avoid overgrowth of microorganisms. Refrigerate within 2 hours. So this is kind of the rule 2 by 2. Refrigerate within 2 hours and eat within 2 minutes. Within 2 days. You see here, on your slides you see that you have, so these are details of these ones. So I kind of extended every single one, so on the farm and at home. So it's for your knowledge. So I described what was yours, HSSP or HACCP, etc. So this was for your knowledge, but everything was summarized here. So why do we control this? Yes, we have to control it. Why do we control it? So just to avoid food waste, for the cost, either cost of medication, cost of stopping work, and cost of different costs. We control just to preserve the nutritional value and to avoid risk for foodborne diseases. So this is why we need to control. How do we control? So as I said, the best can be to avoid, to reduce the AW. So usually we cannot control everything, obviously, but there are 3 main factors that we can play around, or that are easy to control. Composition, we control the composition. AW, so this is one of the easiest things that we can control, so we reduce the AW. The pH, so for example, we decrease the pH, so then the pH is not favorable for microbial growth. Extract outside the food. Oxygen, we play with the oxygen. We play with the temperature. Temperature is just refrigeration or freezing, relative humidity, and atmosphere. So, if we want to remove microorganisms from food, so there are different ways of removing it. Okay, here when I talk about removing, I'm not talking about the one that it's too late. The food is already spoiled. So here it reached the maximum. Here there is nothing we can do. We just throw the food. I'm talking about initial contamination. It's like just the beginning that we can control. If it's too late, then it's too late. There is nothing we can do. Then removing microorganisms, either by filtration or by centrifugation. So we remove filter, so there are kind of pipelines, and they have holes. Usually these holes on the pipelines, they have 0.2 to 0.5 micrometer. So when the liquid goes, for example, juice or even milk, so it goes, the liquid goes out, and then the microorganisms are retained inside. So this is how we do the filtration. Centrifugation, we just centrifuge, and then separate by gravity, we separate the microbes. High temperature treatment, so this is something common. We pasteurize or we sterilize, canning as well. So we put food in cans, we close the can, and we expose the can to heat treatment. Low temperature, if we want to refrigerate and we want to freeze, and then freezing refrigeration doesn't eliminate the microorganisms. So some of them may be eliminated, so they may not resist, but not all of them. So when we freeze, when we freeze meat, it doesn't mean we're killing all the microbes. So we still have microbes that are present, and they can start growing and even overgrowing when we defrost the meat. How do we reduce the AW? By dehydration. So we remove water and we increase the solute concentration. So we do it by drying or by lyophilization. Lyophilization, this is what for example, we make milk powder. So milk powder, so it's we get rid of the fat, and it's exposed, so it gets into like cylinder. It's kind of a spray. So the milk is sprayed inside the cylinder, that has hot air, and this hot air, when it's in contact with the droplets of milk, this is why the droplets get dry, and this is how we form the milk powder. Chemical-based preservation, so when we add additives or specific antimicrobial chemicals, and these are usually they're named or regarded as safe. They're GRAS, so they're safe to be consumed. Then we have microbial-based products, and then here for example, we have microbes that they produce antimicrobial components, so that we call bacteriocin. So microbes that they produce components that kill or destroy other microbes, and then radiation or ionization. Okay, heat treatment. So as I said, pasteurization is the most common one. It eliminates non-spore-forming pathogen and it reduces the spoilage. Pasteurization, here you will say, oh, we may have seen this. We saw in this course or this course that the temperature is this and that. Yeah, so the temperature here changes. And this is what we call a coupled time temperature. We need enough higher temperature and enough time for these microorganisms to be destroyed. So this is why, for example, we expose the product 63 degrees for 30 minutes. If we want to increase, we increase it, but we decrease the temperature. So there should be a balance between time and temperature. So this is what we call coupled time temperature. UHT, something commonly used as well. It's ultra high temperature. So here, instead of 65, so the pasteurization, this range goes from 60 to 65 to 75, while UHT should be higher than 100 degrees. So usually it's called instant. Yeah.
For the 63 and 72, those are two separate options, right? It's not like a
Yes, yeah, no, two separate options. So 62 for more than 20 minutes or 72 for instant.
Do you like damage the food quality if you do like?
Yes, so here it depends on the quality, what you are pasteurizing. Yeah, so you prefer, so you would like to preserve the maximum nutritional value. Yeah. UHT, as I said, senior, very high, and 2 seconds, like instant. So usually there are kind of pipelines that go from I don't know how to call it. It's kind of a passage time, passing from one point to another, and this passage time is 2 seconds. So just say how it's very speed. So then milk or other, they don't get exposed for a long time to very high temperature and to preserve. And then usually there are kind of research done. People try different couples from temperature, and then later they check the nutritional value, they check the vitamin content, etc. So the purpose is to kill microorganisms to preserve the nutritional value. Here, as I said. So, and then there are pros and cons. Pasteurization is not sterile. You may have realized that there is something. Here it's sterile, but it has to be aseptically packaged. So they are packaged in these Tetra Pak. They call it Tetra Pak. I don't know if there is another name. You know, like these bricks, and they can be stored at room temperature before they are open. While pasteurization, even if you think that you are eliminating microorganisms, but after pasteurization, they need, for example, milk has to be stored in cold. Just to prove that the pasteurization doesn't mean sterilization. Okay, filtration. Okay, I forgot to say something here. UHT, yes, it's sterilized, but because it's very high temperature, it may affect the content of it may lead to the caramelization. So it may affect the color may change a little bit. So for example, if you use UHT milk, the color of milk is not white. It's a little bit brownish or off-white. And this is due to the caramelization of sugar that is in milk. Okay, next, filtration. Filtration is just, as I said, we use a membrane of 0.2 to 5 to up to 5 micrometer. It helps reducing 100% of microorganisms. It can be kept at lower temperature. There are pros and cons for filtration. What is good for the filtration is that you are preserving the quality of the nutritional value, because we're just eliminating the microorganisms. The cons is that the filtration is costly, it costs a lot, it's expensive, and for example, if we talk about milk, so it preserves the taste of milk. So the milk tastes as if it was raw milk. So it's the same tasting. And then because the consumers for a very long time have been used to drink pasteurized milk, so sometimes going or consuming raw filtered or microfiltered, so consumers do not accept it because it reminds them of raw milk. So this is why it's not kind of culturally is not accepted, and it's more expensive than pasteurized milk, obviously. Just 15 minutes and it's here. So another way of preserving is fermentation. So fermentation, we ferment either foods or beverages that are produced to control microbial growth and a conversion of food component to enzymatic action. So this is by definition what we say. So what we do, why do we do fermentation? We would like to increase the shelf life, to reduce the pH, and do not allow the non-desirable microbes to grow. We increase the nutritional values because usually these bacteria, during the fermentation, they produce components that are farmable, or they increase the nutritional value. We change the texture, palatability, and digestibility. For example, fermented milk or fermented yogurt, etc. It can be more digested or digestible compared to raw milk or non-fermented. So it helps with the digestion. So there are two types of fermentation. There is a wild fermentation or the spontaneous fermentation, and there is a controlled one. So wild fermentation is that, for example, the kimchi or other. So these are the traditional ones. So you don't control. So you just or like fermented vegetables. So people do it at home. We just put the vegetables in containers. You add salt, you add etc., and you close them and you leave them on the counter for a long time. These are fermented. These are not controlled. So microbes that are coming from contamination, they grow together, and then because they have a nice environment, this is how they grow. And then we have the controlled one that we inoculate. We as people, we inoculate purposely specific microorganisms to either produce yogurt, to produce cheese, etc. Here in this example of fermentation, I will be mainly focusing on milk fermentation. And we'll go together and then try to see what are these microbes that are responsible for milk fermentation. They can produce yogurt and they produce other milk product or dairy product. So before, I wanted to give you an overview or reminder of milk composition. So why these microbes grow in milk? Because milk is very rich. So it has a higher percentage of water, we said it. It has a very high content of proteins, sugar, enzymes, etc. So it's a very perfect niche for growing microorganisms. And they have a pH about 6.7, 6.1. Proteins, the main proteins that are mainly involved, the proteins that are mainly involved in the fermentation are the caseins. So what do we know about caseins? So caseins, you have seen this slide, same slide over and over. Caseins are conjugated sorry, I'm getting tired. I've been talking since 9 this morning. So conjugated proteins with phosphate group is stereophyte. So I did it on purpose. I gave it to you, I want you to remember this. So you have kind of the definition. So first, with phosphate group is stereophyte to serine residue. So this is casein micelle, and then we have here the submicelles, and then we have the hair of the kappa, casein peptides, etc. So they form the structure of the casein, and this structure. This is what, breaking this structure by acidification or after the fermentation, this is what makes the curd of the cheese form, and this is what makes the structure of the yogurt form. So it comes from the caseins, micelle casein. And this is an overview about the network. So we have kappa casein that is surrounding, and we have the alpha and beta casein that are here forming. And obviously, because these are phosphorylated proteins, so we have calcium phosphate in them. So this is an overview. Yeah.
The slide before are the pairs on the casein molecules. Those are the macropeptides, or are those the serine residues?
No, these are the so these are the ones making, like here, these are making kind of connections. And these are the ones that we break first when we start the fermentation.
So they're joining the.
They are joining, yeah. They're not. They're part of. This is what they form the micelle inside.
Okay.
These are hairs. They are layers. This is what they form. The structure, the heavy structure of the casein.
And is it just like the serine residue, or is it just like another random residue on the?
But they are mostly the serine residues.
Okay.
Yeah. Okay. So how does this? So why these casein are important? You see here. So there are different proteins, but the caseins are the ones that are important for the fermentation, because we will see why. So casein, they are usually stable. So how we so milk as it is, it's composed of sugar, lactose, main sugar of milk. This is disugar, so like a B molecule. And main is the casein. So casein these are like kind of 3D molecules. They are composing this matrix of milk. We need to destroy this gathering and to bring the casein together to form, rather than having them in a liquid, we need to form a solid. So this is what, or at least to bring them together, this is what we what we call destabilizing the casein. So they are stable, but then this stability can be affected by the pH, by salt, and by temperature. So salt content affects the content of calcium phosphate. So these bridges, they may be, we'll see it in, so they may be broken, and then like disturbed by, by salt. pH. pH can, at lower pH, so these calcium phosphate, they dissolve. And yeah.
Is that why, like, when you're turning milk into cheese or cheese curds, they put like a crap ton of salt in there to
Yeah, it
formulate
has-
the.
Yeah.
So instead, so what it is? Okay. So the salt is because the high salt content, it is stabilizing the casein, so it's turning the liquid structure of milk into solidified by putting together all the casein micelle
Yes.
molecules.
So the salt, it helps what we call stenolysis. So it helps separating the liquid from the solid. So then the casein structures, the solid part of the milk gets gathered together. And so that's why it's easy. For example, when we're making cheese, we ferment the milk. We add a specific enzyme, the rennet, for example. We break this enzyme. So we break these caseins. So we need to break them, and then we add the salt. So this is a common process in cheese making. So we add the salt and we heat. It's like it's kind of a cascade, one after the other. So this is what makes this big structure break and destabilize it. Heat treatment, as I said, because the way the liquid that is in the milk, it gets absorbed into the micelles, and then dehydration. So dehydration, for example, when we were preparing milk powder. So milk powder, yes, it's preserved, but it kind of destroys or destabilize the caseins. Okay. So there are specific. So milk fermentation is easy compared to others, but kind of tricky because it has to break a specific site or specific region within the casein. And this is how it helps the aggregation. So if we want to precipitate the casein, as I said, we first need to break or destabilize this one, and then so there are two ways of aggregation, kind of separating or breaking this structure. We either add an enzyme, the rennet, or the acid, or both of them together. We'll see in a few slides, it depends on what we are producing in the fermentation, etc. So here, we add rennet when we are making cheese. We don't add rennet, obviously, when we are making yogurt. So the rennet is an enzyme that is extracted from the bovine, and it helps, it's an enzyme, so it cleaves at specific, see here, so this is the kappa casein. This is the kappa casein. It's like a cascade of amino acids. This enzyme, the rennet, it recognizes the site of 105, 106 amino acid linkage, and it goes there and breaks this one. So when it breaks it, it releases the two structures, and this is why it becomes stabilized. Then we have the acidification. What does the acidification do? It's just a decrease there is a decrease of pH to 4.6. So this is kind of a common for the lactic acid bacterial fermentation. Then they reach specific, what we call the PI, point or isoelectric point. And this is the pH that can be reached to destroy this specific or structure of a micelle. So the micelle loses its structure, and then the casein starts precipitating. So this is common, for example, when we make yogurt, etc. This is why, for example, when we make yogurt, there is, so it's kind of packed, but there is a liquid that is, yes, it stays unprisoner, but if you shake you check the yogurt, or if you pour it, you will see that it's really. So how this is in general the ways how we do it. Lactose, we have seen this many times. Lactose is the main sugar of milk. So what all this just to tell you that okay, there is a casein, and casein, the principle is to have these proteins of milk, specifically the casein, that can be broken or cleaved into specific places or region, and then to be able to destroy this 3D structure and precipitate them later. How about the microorganisms? So, microorganisms in food, in milk, they have different origins. They can be, so as I said, once produced within the mammary gland, it is sterile, and when it goes. Although there are, at least in human, there are studies saying even the milk is not sterile or there is an interaction, etc. So even here, we don't. There might be researchers saying the opposite. Anyway, so what is the source of contamination? The farm, the animals themselves, the machines. You know, like the one on the top, this one is a tank where milks are stored and preserved before getting transported, etc. And then these ones, so all these together, they create what we call milk-associated microorganisms, and what I'm calling here milk microbiota. Yeah, because it's, after all, it's a microbiota, it's a microbial community. These microbes, they are different. They can be involved in different functions. They're the ones that are involved in food technology, Lactococcus, Lactobacilli, etc. The ones that are health promoting, same as this one, some yeast, Enterococcus. Some, they're mainly responsible for spoilers, Pseudomonas, phages. You know what are phages? So phages are viruses, and they attack these are viruses of bacteria. So they attack the bacteria, and then they're very known. It's commonly found in milk. So we call them bacteriophages. They are very can cause a very huge problem in the dairy industry. Then one causing illnesses, fungi. Fungi coming from the farm, obviously, from the food of the animals, Campylobacter, and Enterobacteriaceae, mainly E. coli. E. coli, these are indicators of fecal contamination. So these are coliform, they have a form, specific form, and they are indicator of microorganisms. So they are indicator of fecal human contamination, etc. These coliform, they can ferment the lactose. So they can ferment the lactose and produce the CO2. We don't like this. Yeah.
Is that specifically referring to the 015787 serotype?
No.
Oh.
No, this is another one.
Oh, it's another serotype.
No.
Okay.
Yeah. No, this is, I didn't say. When it's not nurture, meaning it's not this specific
Oh,
serotype.
yeah.
Yeah. So these ones. So the ones that are bolded here and the line, this is what we call lactic acid bacteria. So these are the ones that are used for producing dairy products, and these are the ones. So what are lactic? G plus is gram positive. So they're not spore forming. They're cocci or rod. It depends, and they grow on lactose. They mainly they use the lactose as a source of carbon, and they produce the lactic acid, or they produce the acid. They can be homo-fermenter or heterofermenter. What do we mean by homo and heterofermenter? Homo-fermenter, so homo-lactic, so one product. So we start from the glucose and we produce 100% of lactic acid. So one product of fermentation. Heterolactic, we produce lactic acid plus the ethanol plus the CO2. So it depends on one bacteria. So this is the definition. So fermentation is conversion of into two molecules, while here conversion of glucose into lactic acid molecule, carbon dioxide, and ethanol. So it's different process. So they do the same. Here the end product is the lactic acid, ATP. Here we have all these end products, and this is what we call homofermenter, and this is what we call heterofermenter. They are different. So Lactococcus is a homofermenter. It only produces the lactic acid, while Nokonostalk, for example, they're used for specific cheese. They produce CO2. So these are the definitions. So what are the functions? As I said, they produce the lactic acid, they produce specific flavor, and they produce alcohol for heterofermented. They inhibit the microorganism because they produce lactic acid. So good for us. These foodborne pathogen, etc. Most of them they are sensitive to the acid. So it's like these lactic acid bacteria. They produce the acid that may inhibit the pathogens or the non-desirable one. So these ones they can be used either as one single strain or as a mixture or combination. So we use one lactobacilli, or we use Streptococcus and Lactobacillus, we will see. Then we have, so usually they're mesophilic, so they grow at around 30 degrees, 35. Some of them, they may go up to 42, but they are around this temperature. So, yeah.
Sorry, simple is just one strain. Is it just one strain or just one species? Right?
So, we will see. So it depends. It can be either one species or one strain.
Okay.
So for example, in yogurt, one species. So there is a mixture of two species. And we'll see in cheddar cheese, there can be one strain or multiple strains. Okay, so what do we need? These are the ingredients we need for the yogurt. Yogurt, wherever you go. You produce it in Canada, you produce it in South Africa, wherever you go. I just gave an example of South Africa. You produce it in Europe, these microorganisms are the same. Otherwise, it cannot be called or named yogurt. So, we need Streptococcus salivarius thermophilus, and for abbreviation, of course, to shorten it, Streptococcus thermophilus. Thermophilus because it resists to the higher temperature. This is why we call it thermophilus. Then we have Lactobacillus delbrueckii bulgaricus. So these two, if they are not put in the milk, the yogurt cannot be made. These are the rules. So this is a symbiotic blend. So they work together in symbiosis. So first, when we make yogurt, we add these microbes and we incubate at 42 degrees. So this is the common temperature, 42 degrees. Lactobacillus is not thermophilus. It's not thermoresistant. So it doesn't grow immediately. Thermophilus, Streptococcus thermophilus, is the first one growing. It starts growing and then it makes the conditions favorable to have Lactobacillus grow. So this is how it works. How it works. So here, so are responsible for the initial pH drop, and the Lactobacillus are responsible for further increasing because there is thermophilus starts, then comes Lactobacillus. And then yogurt mixture coagulates during the fermentation due to drop the pH because both of them, they are lactic acid producers. So this is usually the tank here. So put it, just pour these microbes and let them ferment. So how we do it? So we need first to start with pasteurized milk. So we need to avoid an overgrowth of pathogen, etc. And then, so first we standardize milk. So what does this mean, standardizing? Because usually the industrials, they receive milks from different farmers, so they put them all together, they mix them. Most of the time they remove the fat, and they start pasteurizing and adding the fermentation, etc. Just 5 minutes and we take a break. So standardization, then heating, so it's maybe pasteurization, then homogenization and cooling. So before we start, then we inoculate. So add the cultures, as I said. For the yogurt, it's a must. Otherwise, we cannot call it yogurt. It's Streptococcus thermophilus and Lactobacillus burgerius. Then adding this, and then incubating for 40. I said 42, but it can be also 43 degrees, etc. And then here it depends on the type of yogurt. So either stirred one or the one that we add fruits, etc. So it really depends. On what do you want. Cheese. Cheese is a fermented, fresh or ripened product that is obtained by the coagulation, casein coagulation or precipitation, and waste separation. We have different types of cheeses. I'm not going to like this, but just to tell you. So we start from the same first product, milk, and then we end by having different cheeses. So there are two main factors that lead to making these types of cheeses. First is the casein precipitation or coagulation. How do we make it? How do we precipitate the casein? And the second is at this step, the processing and the maturation or the ripening. And I forgot one thing is the add of lactic bacteria. So depending on what we are adding, we will end by having specific cheese. So I don't know, cheddar cheese, we add lactococcus. For emmental cheese, we need to add Propionibacterium, etc. Okay, so how do we make it? So we need same as yogurt, we standardize, we heat, and we cool, we add starter bacteria. We need something bacteria to start. We inoculate with lactic bacteria. And I said between brackets non-starters because sometimes we add others later to help them in the maturation or in the aging of cheese. We add rennet, this is really for precipitating the casein. Then when it's precipitated, we cut the curd and add the salt and put separate the whey from the solid, and we can put it for aging or maturation, or just pack it as it is. So the basic is just here, just kind of recapitulating what I said. So lactic acid bacteria, what do? They produce lactic acid. Obviously, they lower the pH. What does this help to do? It helps coagulating or assist the coagulation. It helps promoting the syneresis, separation of whey, separation from the liquid, from the solids from the liquid. And it helps preventing the spoilage because of the acid. These lactic acid bacteria, we call them LAB. It's commonly known for abbreviation. LAB, they also produce some growth factor that can be, so this is why I said these can be beneficial. And they can also help produce metabolites that help in specific cheese, have the non-starters. I know you're lost. The non-starters, so there are what you call starters. Starters, these are the ones that start the fermentation. Without these starters, we cannot have a fermentation. And these are for all cheeses. Most of the cheeses, we need starters. It can be lactobacilli, it can be lactococcus. So these are the two starters. For yogurt, we need streptococcus. Yogurt is different. Then it depends on cheeses. So for example, fresh cheese, we don't need the aging or maturation. So after after separating the curd from the liquid, we pack it and we put it for consumption. We sell it. For the one, for example, like cheddar cheese, we are at this step, like day of the production. This is day of production. But then the curd as it is, we need to or incubate it in another at room temperature at 4 degrees to help aging or maturation of the cheese. So the maturation of the cheese is done by the action of other microbes, other lactic bacteria that we call non-starters, because they are non-starters. They don't start the culture. They don't start the fermentation. They come later, and they help enhancing the flavor. So, what's the purpose of maturation or aging of the cheese? Is just to enhance the flavor. So it's a flavor but more kind of acid, etc. That is characteristic of aged or old cheese. And then to make this old cheese, we need lactic acid bacteria, but we call them non-starters. Okay.
Would the non-starter microorganism vary depending on the type of cheese?
Yes.
Okay.
Yes. A lot. Okay. So, lots of text here. So it's kind of just summarize what I said. So start the culture, then use it. So I put a lot of text so that it's easy to even later to revise even for your information. It doesn't have to be necessary to read for the exam. And so the starters, as I said, so what are these starters? So they include lactococcus, they include streptococcus for the yogurt, they include lactobacillus and like this is kind of to recapitulate. We're adding the rennet, and what does the rennet do? Rennet is just help producing the gel, like it helps in coagulating the milk. So formation of gel, destabilizing of casein. I'm just repeating so that it's easy to memorize. And they make them to aggregate. As I said, there are different ways of aggregating the casein and making the gel. So it's either enzymatic by adding the rennet, or by acidification. So we add either the lactic acid, or we make the bacteria produce the lactic acid, and we heat. So, or we make this coagulation by acidifying and heating. So here it depends on what we're producing. If we want a cheddar cheese, we need a rennet, mainly. If we want cottage cheese, we only need to acidify it. We don't have to go through all this process. If we want to produce ricotta cheese, so it's mainly heating and acidification for this. So see, we start from the same starting, but then the process or the details are changing. This is what makes different cheese. And the coagulation, obviously, it's a denaturation of whey protein. Then store and so then we come to the ripening or maturation. So when we talk about maturation, we talk about bacteria that help enhancing the maturation. We talk about N-slab is a non-starter lactic acid bacteria. We call that, it's commonly, for people who work on lactic bacteria, they commonly use it, lactic acid bacteria. These are adjunct. We don't add them. They're not starters. We don't add them. They are either, we don't add them to start the fermentation. Usually, from what I know, they are not really, so we don't kind of control them. So we make, I worked on cheddar cheese. I did my PhD on cheddar cheese, so this is why I keep saying cheddar cheese, cheddar cheese. We make a curd, we cut, and we leave it in a storage room. The contamination coming from the air, it has lactic acid, bacteria, etc. This is what makes the cheddar cheese the flavor specific of cheddar cheese. But there are specific, can be companies, etc. They do it in purpose. So they kind of control the bacteria that are added and had for the fermentation. Okay. So what are these adjunct one? I keep talking about them. So these are non-starters. As I said, so they help enhancing the flavor, etc. The adjunct starter or NSLA, they're not added in all cheese. So as I said, it's very different. Then here there is a case of these non-starters. They can be lactobacillus, they can be lactococcus, and they can be one typical, you don't find it everywhere, it's called Propionibacterium friedenreichi. Propionibacterium friedenreichi. So this is a typical bacteria that we add purposely, or like not really add, but it's present in the Emmental, in the Swiss cheese. This Swiss cheese, it has, if there is no Propionibacterium, we cannot call it Swiss cheese or this one. Why these microbes are added? Because they are a CO2 producer. They had producing the CO2, which make these specific holes in the cheese. So these are made, holes in the cheese are made by specific lactic bacteria called Propionibacterium. If we don't have this, we cannot call it Propionibacterium. And this one, Emmental, it's AOC. You might have heard about this. It's a French. There is no translation. It's Appellation d'origine contrôlée. It's meaning a naming of controlled. Just trying to translate. Controlled names. It's like Emmental. It has to be from this valley in Switzerland, and it has to be produced because otherwise it cannot be called Emmental. And then there are other examples. Okay, we just go. Maybe we should let's take a 10-minute break and then come back for. Any questions? I know it's a lot of information. Yeah. Yes. They can be used. For example, the camembert. You know the camembert layer, the white layer of the camembert? So this is the yeast. It's not lactic bacteria. This is like kind of another example of the non-starter.
It's not lactic.
Yeast. No, it's not lactic. So this is the only example of hey! Oh, yeah.
Okay. So, okay. I think you explained this later. So the heterofermenters just need to make CO2 and lactic acid, whereas the homo-fermenters were just making just lactic acid.
Yeah.
Is it just one molecule of lactic acid, or that part is just like
One molecule of glucose, one molecule of...
a one-to-one?
Yes, one molecule of glucose, it's said it here in the two molecules of acid, and then it's one once you put
1 to 1 to 1.
Yeah.
Oh, yeah, but,
Okay, great. AW. Oh my god.
I should remember this.
I do. Okay, so bacteria grows at high. They want high AW.
Yeah, they need
Okay.
at least 0.8.
At least zero. Okay, at greater than.
Yeah.
Okay. Greater than 0.8. But all microbes are greater than AW of 0.7.
Yeah.
Okay. And what's the difference between microbes and bacteria?
Microbes have viruses and yeast and anything. These are microscopic ones.
So okay, microbes just mean like small stuff.
Yes.
Okay, and so it's like if we're looking at a microbe, and then there's like bacteria and mold.
Yeah. It can be because mold they're kind of they're totally different. They're microscopic.
Yeah. Okay. Is mold like similar to fungi, like RNA?
Yeah. Yeah.
Is fungi below mold or fungi top of mold? Like it's kind of like who's like in a tree?
Fungi and
Ah, right. This bio. I'm coming in. Okay. Got it. So then, that's okay. You said something about field mold. It can grow at greater than 0.7. Bacteria is 0.8. Oh.
the mold, yeah.
It's coming back. If you include that chart on the test, I'm gonna be so mad. I'll look back at the chart. Okay, go.
How are you, Stephen?
And then,
This you go back to your this is the same slide as yours.
yes. Is this on here? The yogurt slide? Casey slide?
Yeah.
Okay, then it's one or the other happening, right?
Yes.
It's either rennet
Yeah.
or, okay, let me just make
It
sure.
can be two. It depends. I said it depends on what you want.
So
It
either,
really depends.
so what do you mean that it can happen? You can do both?
Yeah. This will just enhance, accelerate the speed. Because after all, for example, when making cheese, you first ferment and then you add the rennet. When we make cheddar cheese, we ferment it. We add the lactic bacteria. We grow the lactic bacteria for at least 2 to 3 hours. Then add the rennet, meaning there is an acidification, then the rennet.
Oh, okay. So, for example, if it was cheddar cheese, you did a PhD on cheese.
Sure, yeah, I did a genomic.
Okay.
So I will talk about my PhD in here.
So for cheddar cheese, you do acid, and then
We do fermentation, then rennet. Yeah, we don't do acid. We do acid, specifically added acid for cottage.
cottage cheese.
Yeah.
Got it. And you said that we need to break down and destabilize the casing
Yeah.
before we do we go on to the other thing. So destabilization is the enzymatic reaction or the acidification, and in the case of rennet, it produces a stabilizer, which is the soluble sample. Great.
Lisa, you remember this? See, she was in the same class.
I was there.
This is true that you were here.
So this one first, if we're looking at this chart, the first question is how do we precipitate the casing, and what was the second question? How do we disturb?
How to precipitate the casein and how if we use lactic acid valve. So we how do we accelerate it and then if we yes or no we use lactic acid.
Oh, how do we Thank you. I am done. Thank you.
This, you go back to your
I can explain it to you.
do it, you go back. You see, I copy and paste from last year's slides. It's the same question. Yeah, we try to take it down. Because here, you remember from last year's slides, we make them, these are type of we make them like, we make, so these are the land, we make them type of yeah, so we make the small units more type of and then we can aggregate the shapes more.
Because the thing is, they're all hydrophobic and they're inside the water, right? Or like, the hydrophobic materials want to stick together because they don't want to be near the hydrophilic material. And what I think it's doing is by aggregating it, we're separating out where we're going to all the casing modules are all throughout the build, they're kind of like everywhere. But when we're doing the aggregation by adding a Reddit or by acidification, we're congregating it by like, we're clumping these casing molecules together. And by that, we're basically curves or like shapes or
Yeah, so you need to break clear the good structure, clear the good structure. So you need to break, this with these bridges. To be able to separate them, so you let what is going in, you let everything it's easier if it takes separate it because the way the liquid is represented inside. So you want to break things that are lower outside. Yeah, there are like water. I mean, this is like what what are these cases?
Yes. Your question.
And then what are the processes? Okay, so back again to our second part. Guys, we're starting again. So, for people who have more questions about casing, I'm happy to re-explain it. So we'll go through. I'm happy to explain it again. I am more than happy. I do have slides and transcripts for these. For people who want to know more about these kerosene and who don't remember, the one who don't remember from last year class. So for the one who have seen this before, you go back please to your slides. For the ones, I'm not pointing anyone. So for the ones, you have your email. I left my email. I will be more than happy to share this information with you. Okay? So, for the one who are taking NFS 386 this year, we'll go over the casein when we talk about proteins, so don't worry. But that will be, for the purpose of this class, I will be sharing this with you. So feel free to email me, I will be happy to share this information for you. You never know, maybe you will get this. Okay. So, other examples of fermentation, so wine fermentation. So we have other dairy products, we have bread red is an example. Wine and wine, so it comes from grapes, fermentation. The grapes are harvested, they're crunched, and they are fermented. So the main fermenters of this product. So we have the yeast, and we have other lactic acid bacteria, or what we call malolactic bacteria. So it's another type of product. So malolactic or malolactic acid, and so the malolactic acid they are produced. It's produced mainly by Echinococcus uni. So Echinococcus is the one producing this. And we have Lactobacilli and Pediococcus. So these are the ones. And then the aging is done in specific conditions, etc., to mainly enhance the quality of the wine. So this is what happened. This is kind of a chart. Start with the substrate, and you don't have to remember this. I just give it to you for your information. Alcoholic fermentation. So as I said here, so it starts with the yeast, and we have other lactic acid bacteria. So yeast is Saccharomyces cerevisiae. Saccharomyces is the one. So without Saccharomyces, we cannot call it wine. So it goes through these. You might have seen this chart in other courses. So it starts from glucose, and it gives ethanol. So overall reaction is done in anaerobic conditions. So we start from one glucose, and we get, so it's heterofermentation. And we get N2 ADP, we get ethanol and CO2. For the lactic bacteria, because here, like I'm just repeating, so then we can go remember the steps. Then the lactic acid bacteria.
Sorry, are these artificially added, or does it just happen organically?
So most of the time they are they're not. No, we don't start them, which is kind of different. So no, Saccharomyces cerevisiae comes from, to the best of my knowledge, we don't add them. They're coming from. We may add lactic bacillus, Echinococcus, but the yeast, no.
So could you refer to Saccharomyces as like heterolactic yeast?
Yes, it's a heterofermenter. Yeah. It's not heterolactic, it's heterofermenter.
Okay.
Yeah. So these are, they convert the malate, that is the sugar that is present in the grape, into lactic acid, or malate, or sometimes so when we say acetate is acetic acid, when we say lactate is lactic acid, when we say malate is a malic acid. So this is what happens in this conversion. So we have Saccharomyces, and we have Echinococcus and other lactic bacteria. The beer, there are two ways of fermentation. So we have the Ale beer and the lager beer. So it depends on what type of fermentation, and it depends on the yeast. So the Saccharomyces cerevisiae, it's a fermentation of malt, so it's kind of these cereals. Using Saccharomyces cerevisiae, it has top fermenting. So it's a top fermentation. It's done at this specific temperature. The bottom fermenting, it's by another Saccharomyces cerevisiae. So you see, the same as cheese, we modify. Here, the beer, here I know it's added for sure. So it's lager or the other top. So it depends on what do we want. Yes.
Sorry, go ahead. Okay. So top fermenting, it just means that it's using the first bacteria, and the bottom fermenting just means
This
it's
is yeast.
the second one?
Yeah.
Oh, it's yeast.
Yes.
Okay.
Yeah. Yes. And what else? So we use malt or the barley. So this is a process. So like these are cereals. So we use the malt. So barley, and then it goes from germination, drying, etc. It goes like juice. So then we have this product, and then we ferment, and we get this specific beer. So usually, there are post-fermentation steps. They go through specific fermentation, filtration, specific process to before they get packaged, and usually the beers are pasteurized. So they're pasteurized and they get packaged in specific, while the wine is not pasteurized. Okay, bread is another example of fermentation. So we need for this. And so we need flour, dough fermentation. So yeast, this is the most common, yeast. And sometimes there are yeast and lactic acid bacteria in the sourdough. So for people who use sourdough and bakeries, etc., it can be a mixture of yeast and sourdough. The one baking at home, we use yeast mainly. You can make your own sourdough. So it's like you keep passaging it, etc. Then portioning, baking. So these are microorganisms in the sourdough or in the leavened bread. So Saccharomyces is the common one, and then we add sourdough starters. So these are mainly lactobacilli they had in the fermentation. So for example, the example of this specific bread, San Francisco sourdough bread, it's mainly made by a lactobacilli. It's Lactobacillus San Francisci. I think it was just isolated from this sourdough and this white old San Francisci. And it's preferentially ferment the maltose. So this lactobacilli is kind of targets the maltose that is present. The yeast is responsible for producing CO2 and fuel later on, same as we do at home. Okay. Then, this was an overview about microbes in food. I may have skipped some. I went very quickly on some, so to catch up on time. But please feel free to email me if you have any questions. The ones who need more details about casein, I'm happy to share more information or more explanation. Okay. How about the omics? Do you know what are omics? No? Okay. Omics is for ohm, O-M-E. Everything, overall. So it's the collective and high throughput analysis. So we talk about proteomics, where we are analyzing the protein, all the proteins in the cell or in the sample. When we talk about transcriptome, when we talk about genomics, so these are the omics. So these are high throughput analysis, so we cannot do it one by one. Like it needs a lot of engineering, a lot of data, and it is bioinformatic tools to study either the microorganisms, here specifically microorganisms, but we can study, we can use omics to study the microbiome in the gut, etc. Here specifically, I'm talking about foods. What do we do? I'm talking about in the food industry, in food microbiology. What do? We can use these to characterize and select starters. Now, before in long time ago. So what if you were working in the industry? Industrials, they have collections of hundreds and thousands of strains, and they put them together Now with the omic, so we can have genomic screening, or genomic characterization of these strains to see what is present and what is not present. And based on that, we select the best combination of strains to use as a starter. We characterize microbial communities in food, milk microbiome, cheese microbiome, maple sap microbiome. I'm giving you examples here. That's what we do. Same for the gut microbiota, for example. Sorry, I said microbiome. No, it's microbiota. Microbial. So you should have corrected me. You know what's the difference between microbiome and microbiota? No?
Sorry.
No, you will see it. No, I'm saying it's no, because I know that Dr. Kavelli has this in her class. Oh, maybe you didn't go through the definitions yet.
No, she didn't today yet.
Oh, yeah, okay. So microbiome is an assemblage of microbes. No, sorry. I have been talking since 9 this morning. I'm lost. Microbiota is a combination or a microbial community. Microbes. When we talk about microbiome, you will see it with Dr. Kamel. I even know what type of references she will give you. So it's a combination of microbes, genes, and genomes within, for example, the gut, or within a human, or within the matrix. So it's microbiome, it's all. So genes, genomes, and all the environment surrounding this sample. So this is what we mean by microbiome. Microbiota are just microbes. So microbiota can be within, it's like within the umbrella of microbiome. Here we talk about microbiota. And detection of food pathogen. So here I'm giving you something that I really like. I don't keep talking about cheddar because I like it. By the way, I don't really like eating cheddar, but it has a meaning for me. I don't know, because I did my PhD, I made cheddar cheese in the lab. I mean, it's not something to be proud of. All my lab mates were doing this. Like, I was not the only one. But this is like we're making cheddar cheese slurries in the lab, and then from the cheese we're extracting the RNA, we're extracting the DNA. It's something, and okay. I know I'm running over time, but the good thing in my lab, because it was a food, and we were working with a lot of industrials. And then sometimes, so you go from, like, you do the optimization, you do we'll show in a few slides, and then you say, okay, I did the characterize, you do this, you choose these strains to make cheese. And they make them, and then they send them to you to analyze the microbiota. But they don't only send you one sample, they send you a huge package. So the beauty of it is that we're not only doing science, but you're only having cheese for free. So this is why I like her. So we're having samples. We're having 3 samples because, okay, I'm sorry, I'm running over time. Because this is for your information, for people who are interested in quality control, etc. Because there are ways of doing of samplings. So, for example, there are rules for taking samples. So, for the production, there is a proportion of. So, for example, if you produce 1,000 of cases of cheese, so the case is considered as 1 unit. If you want to sample it, you need to use the case, or even if you only need 10 grams of this to make your analysis. So this is why, it's a matter of sampling. I know it might not be interesting, but I wanted to share this with you. So how we do this? So I'm giving you an example of Lactococcus lactis that is used to produce cheddar cheese. Lactococcus lactis, subspecies Cremoris. You know that there is a genus, there is a species, and there is subspecies, and there is a strain. So these Lactococcus lactis, they're cocci, they're gram-positive, they're homofermented. They can be used in, you asked me about single strain and multiple strain. So here they can be used as a single strain, and they can be used as mixed strain. So different strains put together. In the industry, it is highly recommended and preferred to use mixture of strains. Why? Because, you remember I told you about phages. In the industry, like dairy industry, phages are very frequent and it's easy to contaminate. So this is why it is preferred to have combination of starters and don't rely on one combination, but have at least 10 combinations of starters that they work all together. They make the same quality of cheese. So then, if there is an accident, if there is a contamination of phage, so you get rid of all the production, decontaminate the plant, and then start again with a new fresh one. So this is what the purpose of food strain. And then, because these microbes, they work in collaboration. So one produces a specific flavor, another one produces another flavor. So this is why it's better to have combinations. How does this work? Just imagine, you put this lactococcus in milk, and then you imagine how do they work. So this is, as I said, it's a coccyte. It's a circle. So this is a cell, one cell, like kind of I'm zooming in 1 gazillion times, so that you can see one cell during the fermentation. The purpose is to produce lactic acid, and so these one, they start growing. These bacteria, they start growing. They need to consume the lactose, glucose from the lactose, and produce the lactic acid. So this is the first step. It's not exactly first, second, etc. cascade, but I'm telling you. So we need the lactococcus to produce lactic acid, to acidify. This bacteria, it needs lactose, so lactose is consumed, it produces the ATP and CO2 and lactic acid. It also needs for its own growth to have a source of proteins. The proteins are coming from the casein. See, the casein is important. So the rennet acidification is here. It is needed for bacterial growth, for bacterial fermentation. It needs protein. Casein is a big, huge molecule. It cannot be consumed or uptaken by the bacteria as it is. So the bacteria will cleave it. This regardless of the rennet. And this happens before adding the rennet. So bacteria will cleave it. Bacteria, Lactococcus, has a specific proteolytic system. It has its own scissor. Just imagine, protease that is at the surface, and it cleaves. Can you imagine? So these are the casein, I draw them. It cleaves the casein in small polypeptides. And then these polypeptides, they get translated or transported inside. The reason why they transported, because the bacteria need to grow, need the source of proteins. So this is why they're transported through transporters that we call oligopeptides, OPPs. And so these are the uptake, the transport, the hydrolyzed product. And then once inside, these one you see, I drew 3 small balls, and then they are degraded in 1 unit, 1 amino acid. The bacteria will use what they need for their consumption and their growth, but there are left over of amino acids from the casein degradation inside. At the end of the fermentation, for when we make cheddar cheese, so we have about 1-2 hours of fermentation, then we add the rennet, we salt, and this is what we call heating. So we go from 32 degrees, and we increase the temperature to 39 degrees. 39 degrees, it stops the enzymatic reaction, and it stops stops the bacterial growth. At this time, here, these microbes, you know the stationary phase of bacterial growth? So bacteria, at some point, they are, you can just imagine, they are tired. There is less, they're tired. There is less, I am the one who is tired. They have less food, the pH is too low, etc. So all the conditions are not favorable. Here, they're not growing. They're not metabolically active. They're just here and they want to suicide. Let's put it this way. It's like autophagy. You know the phages of cells? So it's the same as bacteria. At the end of the fermentation, because of all these conditions, they're not, so they start auto-destroying themselves. By destroying themselves, they do we call the autolysis. They release themselves. Releasing themselves, they release a specific enzyme. Here, it comes and starts cutting the membrane. When they release, you see what was released? The amino acids that are left over from the casein. These ones are very important to make or to have, enhancing the flavor of cheddar cheese. You see, it's something like kind of a cascade, and now we know. So these ones, amino acids, can be any type of amino acid. Can be hydrophobic amino acid, can be aromatic amino acid, can be anything. Same thing as in here. So the production, this specific enzyme, when it cuts this one, protein, when it cuts, it has a specific way or specific site of cleavage. So depending on where, and this cleavage is strain specific. So Lactococcus lactis number 1, it cleaves, for example, at site number 155. Lactococcus lactis number 2, it cleaves at site number 62. The differences of cleavage will result in differences of amino acid, and some will result in producing hydrophobic amino acid. Hydrophobic amino acid are the ones that are responsible for bitterness. Did you get it?
So different amino acids give
Sorry,
different tastes?
Sorry?
Different amino acids give off different tastes?
Yes, different amino acids coming from this step and this step are the ones responsible for the taste. Hydrophobic amino acids are the ones responsible for the bitterness. Yes?
Just a question. That was like 3 types, or those 3 types of casein or 3 types of PRTP or?
No, so 3 types of specificity of the casein.
Okay.
Yes.
From casein to the strain specific PRTP.
Yes.
Okay.
So this one, PRTP, this protein is that cuts the casein in, so then they can enter the cells. These are specific. They can be classified in 3 classes.
So
Yes.
it's like cleavage sites, basically?
Yes.
Okay, thank you.
They have different cleavage sites. And then this specificity or this cleavage site comes from the genetic diversity or variability of the strain itself.
Okay.
Because genetic diversity may lead to a variability of amino acid and may lead to a variability in specificity of the enzymatic reaction. Yeah.
Are there also peptidases that leave at different positions in the
No, the peptides are not that specific. So so all is done by the PRTP.
And what triggers the autolysis?
Is the conditions, are the environmental conditions, are the salt, heating, and the pH.
Okay, so that's all.
Yes, yeah, yes. It's like a autophage. Okay? So, as I said, they produce bitterness, and this is what we do. These Lactococcus lactis, so different produce. So these Lactococcus lactis, they have the production of cheese is strain specific. Different strains, they have different technological and sensorial attributes. So they don't produce the same quality of cheese from one to another. There is a difference. The reason why they are different, it's not only because of the PRTP, it's overall because of the genetic diversity or the genomic variability. So here I'm giving you examples. These are strains. For example, this one, if we use it, it gives us, it's a good one. This one, these two, the quality is so and so. This one, the quality is not that good. Plus, it has a PRTP that produces a bitterness. Okay, so as an industrial, you don't want to only rely on the PRTP to say, oh, it cleaves, and if this one doesn't cleave, etc. You want to have a full screening of the genomes and to see, because the genomes, what the genomic analysis gives you, it tells you what genes are present and what genes are absent. Gene present meaning function, gene absent meaning there is no function. So if there is no gene coding for the PRTP, meaning there is no function of the PRTP, and so on. So this is what you do screening. The screening, genetic screening, it helps you predict the behavior of these strains within, during the fermentation or during the thing. So genetic diversity is known as an important reservoir. So comparative genomics. So if we want to see why this one is good and why this one is bad, so what we do, we just compare the genomes and we see what is it present in the good one and absent in the bad one. So this is what we did. So this is kind of part, when I started my PhD, this is what I did. I had to do genomic screening of strains and to see which one was good and why they were good. So so this was, we call it the genomic comparative hybridization. It's just to get like screening. So what you do, you extract the DNA and then use specific tools to do hybridization and to identify. So this is a heat map. You know what is a heat map? So a heat map, so showing in columns, every column shows one strain, and every line shows one gene. Bacteria, they don't have the genome of bacteria. It contains about 2,000 to 2,500 genes. So it's not like human genome where it's huge. So this is what the genomic comparative gives us. The genomic comparative tells us what is present and what is absent. And from the absence and present, we'll see what strain we can select and what strain we can exclude for the cheese. So for example, so here, for example, so this is the heat map. Black means presence, gene presence. White meaning gene absence. And gray in the middle here, meaning the gene has a lot of steps, single nucleotide polymorphism. So in this polymorphism may lead to variability in the gene function. So when you see this, we have to say, okay, the gene is not that functional. We have to be careful. So, for example, if we look at the osmoregulation, the gene coding for the osmoregulation are the gene involved in resistance to salt. So it osmoregulates. So for example, if I have genes that are absent, I know that these strains, they don't resist to the salt. So is it a good or not? Same for the lysis, same for the phages, etc. So this is how we do screening. So we identify genes absent and present, and then based on this identifying, we can predict the presence and absence of function. Okay. So this kind of was a conclusion. So genomic comparative, it's a tool that helps estimating the diversity among the strains. And so, for example, in this project I was working on, so what we did, we had, so it was a collaboration with dairy farmers and different cheese producers, and we had to do screening. So we had to work on hundreds of strains, and then do the screenings of every single one, and then select the one that have the genes, for example, that we need, and then use a combination of them. And they say, okay, you can use, we have strain number A, you can exclude, strain number 100, you keep, strain, so this is what is, so this is how we use the tools, genomic tools in this industry. It has identified genetic biomarkers, the example of osmoregulation, so we could identify any others. So in the industry, we use the genomics or the omics to relate genetic diversity to performance. It's diversity meaning performance, meaning they can do the function or they cannot do the function. They can behave this way or differently. So it has selecting, as I said, according to their genomic profile. The omics had predicting the strain's response. So the osmoregulation, for example, we know that this one, they don't resist to the salting step when we add salt to the cheddar cheese, etc. Is this enough to screen the strains? I would say no, because functional gene, presence of gene, meaning yes, there is a function, but is this a function done at 100%, or it is not, or the gene is somehow inactivated or not transcribed properly? This is what. So this was a good screening, but what needed to be done was to go over an adult omic to study the transcriptomic. The response, the entire response, the gene expression of the entire strains in response to the salting step, in response to the heating, etc. So this was another. How about, we're almost done. How about milk-associated microbiome, milk microbiome, or microbial community? So milk, as we said it, is not sterile. There are microbes that are specific. There are artisanal cheeses that are produced by non-heat treated milk, and they have this specific flavor characteristic to these artisanal cheeses, and this comes from the microbiota. So as you can see here, this is a kind of a combination of different papers, and it tells you how the microbiota or the microbial communities can change from one season to another. You see here, spring, summer, and fall, and the microbiota can be influenced by the weather conditions. And you see here, so for example, these are the years. Okay, so this is the chart. They show the alpha diversity. You will see alpha diversity with Dr. Komeli. Alpha diversity, just remember, there is, so these are microbial, microbial comparison. When we look at the microbiota, we look at the alpha diversity. Alpha diversity is the diversity within a sample. How diverse or how rich is a sample. Beta diversity is between the sample. How different are samples one from each other. So just remember, there is an alpha diversity. I won't go through this because I was supposed to have this lecture in October. So, and then switch it, and I just kept it here. So here is, we measure the alpha diversity using observable OTUs. You will see this with Dr. Komeli. Observable OTUs, OTU is operational taxonomic unit. This is a unit that it gives us an idea about what is present and what is absent, and unit fact distance. So it gives you an overall idea about if, for example, here it tells you that at the spring, the microbial community is more rich compared to the summer and the fall. See, box plot of the summer and OTUs. So OTUs are more observed. So the microbial community is rich in the summer, in the spring compared to summer and the fall. Same for here. Here, this shows you different bacterial phyla. One phylum, different phyla. Here we have Actinobacter, Firmicutes, and Proteobacter. And you can see their level changes over time. These phyla. So it just tells us, same as the microbiota. If you will go through this course about how the microbiota changes with the diet, how it changes with the disease, and how it's disease associated with the microbial dysbiosis, etc. So you just imagine this, we extrapolate it to microbes in food. So it's the same principle. Here, same thing. When we look at the microbiota, we see that there is a difference between raw and pasteurized milk. You remember, pasteurization destroys microorganisms, doesn't destroy 100%, it keeps some of them, and we can see here the difference between these. So why do we do this? We do this because it's important for us to know what microbes do we have when we start making a dairy product, either cheese or yogurt. What do we have in the beginning? Because the baseline we have in the beginning may significantly affect the quality of the product at the end. So this is why it is important to characterize the beginning. Then here another example of cheddar cheese microbiota and metabolome. You see here, so this was from almost 2 years ago. And then, so beta diversity, the diversity between the sample. Here, this plot, it tells you the diversity between, so every dot is one sample, and then what they did in this study, they wanted to see if there was a difference between cheese that was made artisanal, or traditional one, and the one that was made in another. And then they had different productions, and they compared. As you can see here, the artisanal, they're all clustered together, and then the industrial, they're all clustered together. This means that the microbial community are different from the industrial to the artisanal. Same for here. So they looked at the core and the surface. So, and they wanted to see what was the difference between the core and the surface for cheddar cheese. So this is, you will see this a lot in this course because it's a microbiome course. But this, what you will see will be applied to human microbiota or human microbiome. And here I'm giving you the application of this omics. We move it from plant science to food industry. So that's how things are done. Last slide, I want to share. Okay, so the last slide is this is something done in the lab where I did my PhD. So it was looking at the microbiota of maple syrup. How to say? Did you ever think about it that maple syrup might have its own microbiota? No, right? We always think that, oh, maple syrup may be sterile. So, yes, maple syrup is supposed to be sterile. But the way how it is collected, so there are different ways. So either the traditional way, so they have this bucket on the tree and they collect, or they have these tubes. They're connected from one tree to another, and then from all the trees they're collected and put into a can. So here, for example, so the sap, maple sap, is collected here. So this is the traditional, or they can be pipelines or pipes or tubes from one tree to another. And then they put all together, and then they are concentrated or submitted to heat treatment before we start consuming it. But they found that maple syrup, the darkness of maple syrup, the quality of maple syrup could be associated to the initial microbiota. So from where these microbiota or microbes come? They come from the containers. This one, because they're not sterile, they're exposed to air, and they come mainly from the tubes. These tubes, because there's nowhere to sterilize them, of course, they can form inside. They can be contaminated by mainly gram-negative bacteria, and they may form what we call biofilms. This kind of a combination, if a film that is produced, that is made by bacteria. It's kind of a connection of bacteria together, mainly gram-negative, and this is how so the biofilms are made. And these biofilms, they may be responsible for producing specific metabolites, and they can definitely change the grade of the syrup, maple syrup, or the quality or the flavor. So this is what was done. So Marie was the one, so they collected maple syrup from all from maple syrup producers in Quebec, collected them, extracted the DNA, and looked at the microbiota the same way as we look at the microbiota in human. Fecal microbiota, etc. Same way. Principle is the same, etc. It's just the starting sample is different, and the interpretation is obviously different. So, but then the omics, the principles, the techniques are the same. When we run bioinformatic analysis, either we run it for maple syrup cheddar, or we run it for human samples, the principles are the same. So we found, why you say we? So what they did, so they found what was one of the typical characteristics of these microbial across this one, is the presence of Pseudomonas. Remember, Pseudomonas is it's everywhere. So it can be a source of contamination from a farm or something, and it keeps going over and over. And in this study, this study was one of these two were one of the first investigating the association. First, looking at the presence of microbes or microbial DNA in the maple syrup and associating this to the flavor and the color of maple syrup. When we study the microbiota, you will see, I don't want to extend it, but these are based on bacterial DNA profiling. So we don't really know if microbes are present alive or not. So these are mainly based on DNA extraction. But the DNA doesn't mean that microbes are alive and active. So here I gave you an overview about how we use the omics in food, mainly like food processing or food industry. I'd be happy. I went very fast to be able to be on time to over some slides. I'll be happy to answer your questions and give you more information. Feel free to email me. This is my email address. If you have any questions. Do you have any questions? I know you were just want to go. You are tired, I know.